way through breeder training and adoption. There probably isn't another person around with the wealth of knowledge that Guillaume has inside and out about BreeDBase. Prior to joining Lukas's group, he did a PhD with Matilda Klaus at INRA, and he brings industry experience from working at both HM Klaus and Syngenta Seeds. In his spare time, Guillaume is an avid runner and a scuba enthusiast. Okay, Star.
Yeah, thank you very much, Maura. So, Guillaume, would you like to introduce your co-presenter, Lukas?
Okay. Good morning, everyone. I'll try to introduce Lukas. Where to start? Well, Lukas is our team leader. He's been working at GTI for, I don't know, a decade, 10 years about. Before you were working at Stanford University. And Lukas's work is focused on database development and bioinformatic tools. He initially developed soulgenomics.org, which is a portal for Solanaceae genomics. And from Solanaceae genomics, there was a large incentive to develop breeder-oriented applications. So Cassava base, among other databases, were developed in the past years. And that's where the origin of BreeDBase, which will be presented by Lukas today, is rooting. Thank you all for attending.
Okay, thank you for the kind introduction. And I'll just briefly go and give an introduction to BreeDBase, where does it come from, and go through some simple use case for the phenotyping, and then Guillaume will cover other aspects of the database. And also, I think we'll give a little bit of a live demo as well, mainly in Guillaume's part. So this is just the overview, so it's going to be a long presentation. I'll give some intro, then talk about the phenotyping, as I said, barcoding, Fieldbook app, and then we go through the genotyping sampling procedure, and also crossing, and intermixed, I think, interspersed in the presentation is also some analysis that BreeDBase can do. So where does it come from? Well, we have been developing this website called soulgenomics.net for a long time. We started in 2003 in Steve Tanks's lab, and that's a database for tomato genomics, and also served as the main portal for the Tomato Genome Project. And we thought that genomes are not really interested in an isolated way, so we want to compare them to other genomes to be more meaningful. So we made a database that contained all of the Solanaceae plus other reference species such as Arabidopsis as well. And so all these species could be compared in this database. And very early on, we started to be also interested in describing phenotypes. Phenotyping information is really important for a lot of biology, especially if you want to identify genes using technologies such as QTL or GWAS. And so we started describing phenotypes. We created the first ontology for tomato biology. It was one of the very early, actually, trait ontologies that was defined here in the lab by Nama Menda, who came from Daniel Meer's lab. And because we had this genomics and phenotype rich database, I think it was a perfect starting point for a breeding database, because what is breeding, especially molecular breeding and genome-based breeding? Well, you need a lot of phenotypic information, you need a lot of genotypic information. So that basis that we built with SGN was perfect to leverage into a breeding database. And we applied it first to a database called Cassava base. And Cassava base is the NextGen project database, and essentially has the goal of implementing genomic selection for cassava. And so we have huge amounts of information, genotyping and phenotyping information in this database. I think we have around 5,000 phenotypic trials with 11 million phenotypic measurements in this database, as well as I think 32,000 genotypes. And from there on, we created other sites such as YAM-based, sweet potato base, and Musa-based, which are all root and tuber crops in the CGIAR. And they all work in a similar way to Cassava base. And we created other sites such as citrusScreening.org that deal with different problems, such as citrus screening disease. With citrus screening, we have a database of fern genomics and so forth. So we have used this SGN infrastructure to create a lot of different sites. And they all work in exactly the same way, although they can have different user interfaces. So we can customize the user interface. This is just to give a little bit of an overview over the breeding cycle. Probably most of you know that. So you do basically crossing, evaluation, analysis, selection, crossing, and so forth, and you go around that cycle. And BreeDBase really assists you in every operation in this cycle. So what we want to create is a digital ecosystem where any type of data that you use during breeding is managed by BreeDBase. This is just a slide that I stole from Jean-Luc Yannick that describes genomic selection. So if you have a lot of genotypic data and a lot of phenotypic data, you can use modern breeding approaches such as genomic selection, where you have a model training cycle on the left, where you use phenotypic data to correlate with genotypic data, and you build these statistical correlations, and then you can use the correlations in the line development cycle to use genotypic information to predict the phenotypic information. And that results in a speed up of the breeding cycle because you don't have to go out and phenotype. You don't have to wait even until the phenotypes appear. For example, in Cassava, the root grows quite slowly, and you can cross way before the root is really grown. So that speed up is the main reason why genomic selection is interesting, because you get compound interest with every cycle. So what are the challenges in breeding and in data management for breeding? Well, in the old days, people used Excel, and they still do. I don't think it's too old days, actually. And Excel, of course, Excel is was a revolutionary program. It is very useful for a lot of things. We all appreciate Excel, but it's not the ideal tool for data management. So what are the issues? Well, the issue is that your data might be distributed on different laptops that can fall off some desk and disappear. It's very hard to query Excel sheets in aggregate. So if you have different information in different Excel sheets, how do you make a query on in both files? That's almost impossible. So you have to resort to copy and pasting and so forth. But it certainly has a number of benefits. It's easy to use. People have control and so forth. So it has definitely its benefits. But I think the long-term liabilities are for a larger breeding program are really bad. So what are the objectives then in BreeDBase? Well, what we want to do in BreeDBase is that every action that you take in a breeding program does what we call database round tripping. So every action really has to start with the database and not in some Excel sheet. And what then happens is that you export data from the database and you do stuff, for example, collect data, and identifiers are embedded in what you have exported, and new information is associated with identifiers, and then go back into the database. For example, one good way of getting identifiers out there that the database knows about is barcoding a field, for example. So you can just read a barcode, and you know that that identifier exists in the database, and you can associate information with it. And so that's the way all the operations that are implemented in BreeDBase are based on. And so we heard about SGN and Cassava base and Musa base and all these different bases, and people were very confused because we were always referring to that infrastructure as Cassava base, and people were wondering, well, what if I have a different crop? Can I use it as well, or can only Cassava data go into this database? And of course, the answer is no. We can create instances for any crop, and that's why we came up with this new name and the tentative logo, and we call it BreeDBase. And so BreeDBase implements all of our experience that we have gained with SGN and Cassava base and the other bases, and it's really based on a number of very important technologies. And I think the most important thing I want to say is, if you have a database, you have to use standardized data descriptors. Okay, that's the most important thing. Then BreeDBase also allows you to manage fields and planting materials, and it supports the digital ecosystem. So, for example, it supports digital data acquisition, mainly using apps, and that covers phenotyping, genotyping, and crossing. And then it has, once the data is in the database, it has analysis tools that allow you to run analysis right in the database. So you don't need to export the data to do your common analysis, but it also, of course, allows you to export the data and do whatever you want with it. So briefly about the standardized data description. We are basing the data descriptors on something called crop ontology, which is the main, body of standardization for ontologies in the CGIAR. It's run by Biodiversity in Montpellier and I guess Rome, and they have defined, together with the breeders, they have defined a large number of ontologies for different crops that are important in the CGIAR. And they have a website, and that's a screenshot from the website, and you can go there and you can download. So what's an ontology? An ontology is basically a controlled vocabulary, but the terms have different relationships to each other. So this is it's a structured vocabulary, and I don't want to go into the details, but this is kind of a little ontology that describes pizza toppings. But of course, you can describe much more complicated things, such as traits of a plant. And of course, these are vastly more complex and have hundreds of terms. Other ontologies, such as the gene ontology, are even way more complex than that. I think gene ontology has about 30 or 40,000 terms. So you can imagine that it gets quite complicated to maintain the ontology, and certain rules that the ontology must follow. For example, you see that the terms have relationships to other terms, but they have a direction. But it's not a good idea to have cycles, for example, in the ontology. And there are other rules that need to be followed. And this is just a screenshot of the Cassava ontology that we have worked with for a long time, and that we developed together with our colleagues at IITA, such as Afolabi Agbona, Chidozi, and Egesi, and Robert Favucci, and all the other leaders that are involved in that project. And on this side, Guillaume and Nama have been working heavily with the ontology. So let's go through some of the in this cycle. So as we said, breeding is basically this cycle of making selections, crossing, evaluation, and analysis. I think in the next few slides I will be talking about evaluation, and then Guillaume will be more talking about, and maybe a little bit of analysis, and then Guillaume will be talking more about the crossing and evaluation in terms of genotyping. So I will focus on phenotyping. So the database allows you to do all that, and so evaluation, of course, will include activities such as searching data. You can search trait data, trial data, germplasm, and so forth. We also have a powerful wizard search, which I think we can maybe briefly demo at the end of the talk. I don't think I have any slides on that, but that's a key tool that we have. Then it also allows you to manage data. So managing data means that you're actually adding data mostly, and then working with the data in the database. And it can be things like, I'm designing a trial, and for that you need other kind of structures in the database, and the database helps you with that. Such as we have a tool called the list tools, and we have a tool called the dataset tools that allows the breeders to maintain lists of important lines, for example, or traits that then can be used to design experiments and in other tools as well. Of course, we also manage location data. So we have a powerful location management tool. It works nicely with a map interface. And then you can do analysis. You can get summary statistics of your data, of your field, of your experiments. You can build selection indices and order lines according to your selection index. We have ANOVA. You can compare trials to each other. You can do the genomic selection method using a tool called SolGS. And we also are developing a SolGWAS tool. We're also developing a mixed modeling tool. And we have extensive tools to manage genotypic information and to visualize genotypes on JBrowse and so forth.
Can I just ask a quick question? It's Gary here.
Yeah, hello.
Hi, how are you?
Good, how are you?
Good, thanks. Are you going to talk about how these tools may be co-developed or linked in through BreeDBase with other databases?
Yes, a little bit later on in the talk. I will talk about that.
Because there are a lot of mixed model tools around already in different places.
Okay, yeah, I'll talk about that in a few slides.
Thank you.
So one way the breeder has to interact with the database, and so how do we make it as easy as possible for the breeder to know what he or she has to do when he or she is on the database? And how this works in BreeDBase is that we have so-called workflows, and in a workflow you get a series of windows essentially that ask you questions and you enter information, and in the end you have maybe set up a trial, or you have generated a genotyping plate or some other activity on the site, but it's very easy to use. This is what it basically looks like. So we have these different steps that they're on top of the workflow, and the different steps are in gray, but as you move through them, they become green. And at the end, on the right-hand side here, you're done with your workflow. And so this is, on the top, you see the design a new trial workflow, and on the bottom, you see the upload genotypes workflow. And of course, underneath of each step, you have some form that you have to fill in, and it will validate the form and then move to the next step. Another important thing that we have done, I think, is we interact heavily with apps that do the actual data collection in the field, for example. And I think it has been an extremely fruitful collaboration with the group in Kansas that develops the Pheno apps. They have a long list of apps they're developing for any aspect of breeding, and it dovetails really nicely with what we are doing in BreeDBase. And so we want to support all their apps essentially in BreeDBase. And so far, we have been working a lot with Fieldbook, with the coordinate app, with the inventory app, and now we are also implementing the intercross app. So that's been a really great help for us to be able to make these tools available on BreeDBase. So just briefly to show you, and this is not an interactive demo, but we can maybe do that later if you have questions about the process. So this is the design, the field layout trial, and you can see what it looks like now when you see the entire screen. This is design a new trial. You see the different steps that it takes, and this is the first input mask that you would have to fill in, or the second, actually, the second. The first one is a help screen that explains the entire process. Then you see the second circle there is a green circle is green, so that means that's your current step. If full green circle means it's a completed step. And you enter all this data, and then you move through the layout, and you basically have a new layout in the database at the end of the process. And that's what it looks like. You go to the essentially you created the trial, and each trial is a trial detail page, and you can see all the information about the trial. And here, actually, in this section of the page, we see the trial layout viewer, which shows you the layout, the blocks, the even blocks, the odd blocks, the rep numbers, the checks, and so on. And you can mouse over this. You can overlay. If you have collected data already for the trial, you can mouse over, and you will see what data you have collected. You can get a heat map on this of all the traits. And we're working on actually a new version that overlays this information on Google Maps with actual geo-coordinates. So that's something that will be coming up hopefully soon. This is a super functional and easy to use tool. That's very nice. A very important thing is that if you want to stay in this digital ecosystem, is that you should barcode everything in the field. And here we see some examples of barcoding. On the upper left here, I think that's sweet potato. On the right here is this banana. And I think this is actually a yam example. And so BreeDBase makes it extremely easy to barcode fields. We have a label designer. You can design your own label. You can look whatever you want. You can use 2D or 1D barcodes. You can then customize the label to have maybe the accession name, the plot name, and maybe a pedigree on there, and the plot number. And you can print these on a variety of different labels. We support laser printer labels, which are very popular because they're very cheap. But it also supports the Zebra printing language, so you can print to any Zebra printer that's out there, which are very popular as well, and which also have a lot of different kinds of labels, such as sticky labels and so on. Another thing the database can do is track seed inventory. It tracks seed lots, and these are maybe a misnomer because seed lots in cassava and yam can also be not seeds but roots and stakes. So we can track anything you plant. And of course, these are also available as barcodes, and you can track the storage location and the quantity that you have available either in counts or in mass, in grams or milligrams. So how do we do the phenotyping, the data acquisition? I've mentioned that we have worked with the Kansas group, Jesse Cohen's group, who has created the Fieldbook app. And so essentially, that's our main tool for acquiring phenotypic data. But there are other tools such as drones. We are working on interfacing the database better with drone data. What's very popular now is Neer's data. That's Neer infrared spectroscopy data, which allows you to measure a vast variety of properties in your material for very cheaply. Once everything is set up, it's not easy to set up because you need a lot of calibration. But once that is done, it's actually very cheap and fast to get a lot of different data from your samples. Another thing that we are interested in, but we haven't worked on it as much, is ground penetrating radar, which seems something that might be useful for root and tuber crops. So just briefly about the tablet. So essentially, the database allows you to export trials to the tablet in a very easy way. You dump a file and you copy it to the tablet. And you also need to get a trait list, so you can do that as well in the database. Dump this data, copy it to the tablet. You can then take the tablet to the field, and fields in Africa and elsewhere, even in the US, I think many fields are huge. They often don't have network availability, and so with the tablet, once you're out in the field, you're completely independent of the internet. You don't need any connection. You can collect your data. And when you come back to your lab, you can upload the data to the database. This is just a page from the Google Play Store. The Fieldbook is available for free. It can be installed with a click of your finger, basically, and you know, it's available right away. So this is the data flow. So essentially, what you need to do to be able to collect phenotypic data, you need to create your trial or upload at least your trial into the database, and of course, plant and barcode the field. We allow actually phenotyping at the plot level or at the plant level. So if it's at the plot level, each plot gets a barcode. If it's at the plant level, each plant gets a barcode. Well, of course, you can do mixed things. You could have a plot barcode and plant barcodes and collect both types of data simultaneously. And then you export the data to the Fieldbook app. The Fieldbook app supports the barcodes that we are generating. And it can, when you scan the barcode, it will immediately position you for input on that sample. Then you collect the phenotypes, as I said, and you upload it back to the database. And you see that round tripping that I was mentioning before, that's implemented here. So it comes from the database, you do something, and it goes back into the database. And that's key. If you don't do that, it's very hard to manage data. And here you see an action shot of people in the field who are scanning a barcode and then entering data. So now we'll move on to genotyping samples acquisition. And I took exactly half an hour, which is amazing. So I'll hand over to Guillaume, who will present that part. And thank you for your attention. And we'll talk about BRAPI later. This is not over yet.
Thank you, Lukas. Maybe before proceeding, do you have any burning question you would like us to address? Any question? No?
Yes.
I don't see any message.
I have a question.
Oh, yes.
Yeah, this is Rajaguru calling from ICRISAT. So
Hello.
my question, yeah, this is Rajaguru from ICRISAT.
So hi, Rajaguru.
Yeah, so it's a wonderful initiative of this BreeDBase. So my question is like, so there are some efforts going on in terms of databases like BMS kind of thing. So how far this is a difference? How do you differentiate with the existing tools like BMS integration of acquisition of all this phenotypic and genotypic data kind of thing?
Yeah, so at least initially, our differentiation was that we do, that the database supports high density genotyping data and can implement the genomic selection paradigm, which I think BMS, at least at the time, it wasn't a goal of BMS to do that, and I'm not 100% sure if that feature is available now in BMS. So I think that is the big differentiator between the two systems.
Yeah, okay, thank you.
Just on that, my impression, my understanding was that you were not able to handle high-density genotypic data as well, that this was going to be essentially done through links into GOBii. Is that not the plan? I mean, that's not the primary differentiation between these two systems in the minds of the people who are paying for them.
Yeah, yes, our plan is to support GOBii as a backend, and we're working hard on that. The system supports high-density genotyping, but not to the same scale as GOBii. So for small breeding programs and smaller programs, you know, I think our solution is adequate. But if you go to larger programs, such as, you know, I think Cassava will very soon be in that area. We need a GOBii backend, and so we're working to integrate the GOBii backend into the platform. but it will be mostly to the user. Will be mostly transparent, I think, that they use a GOBii backend. That it will just be faster.
Yeah, I think the honest answer to Rajaguru's question is there's not a lot of difference between the capacities of these systems as they've evolved. They are really quite parallel.
Yeah, exactly. I think BMS it's a moving field. It's very fast moving. Breeding technologies evolve fast, and all the systems I think try to adapt. When we started out with NextGen, there was no system that could do what NextGen needed to do, so we implemented that. But I think BMS has been catching up, and so.
And EBS. Yeah. So there's a number of other.
Right.
There's other. Yeah.
Yeah, Rajaguru, BMS also has an upcoming webinar demo, so stay in tune. Then you can learn more yourself to see the differences.
Yeah. Thanks everyone. Yeah.
September or October.
Yeah. Thank you.
Sure.
Okay. Thank you for the question.
Guillaume, go ahead.
Okay. Thank you. So I will follow up Lukas' presentation with similar components but different data types. So what does that mean? Essentially, I will cover now how the database, in conjunction with the PhenoApp, deal with genotyping data acquisition. So the scheme looks similar to the one shown for Fieldbook, and it has reasons to. It relies on the exact same principles where you expect the user to create an experiment in the database, generate the subsequent barcodes, and again, use them to coordinate, as the app is called, the information from the field to the tissues you will sample from the field to the lab. So basically what coordinate is in charge of is to link the identifier of a given plot in the field to the samples you get from that plot into a PCR plate that you may want to ship for genotyping or sequencing at your provider. Solution provider for those technologies. So again, once the data is acquired by the coordinate app, you want to upload it back to the database. And from the database, you can either download a sample format, which corresponds to different providers' requirements, such as .seq or Intertext. And in the near future, we hope to have also a direct, an ability to send directly through the web the form to these providers. There are further details on those aspects available at these URLs if you want to have a look, including some tutorials. Finally, in terms of data types, in breeding, the other big element beyond phenotypes and genotypes is the actual cross activity that is performed to generate variability. This component is new and under development, and takes, again, a similar approach, combining an app called Intercross, developed with PhenoApp's colleagues, and the BreeDBase implementation of this tool into Cassava base. So technically, same process, going to the field, linking a male plot to a female plot when you perform an actual cross, with a slight difference where you can print as you go the labels in relation to the cross you perform. Once you perform the cross, of course, you will wait to get seeds, hopefully, and then you harvest your seeds. You can also collect related cross data with the app and bring it back to the database again. As I said, this is not yet available. There is a beta version of the Intercross app on the PhenoApp's website, as well as ongoing developments for the BreeDBase components of this tool. Just added this to illustrate what kind of work we are at the moment. Currently, many programs would deal with this type of solution, which is basically the recording of the obtained seeds from a given cross in the field using a small tag, and loading those seeds in a small envelope that will be stored in a seed repository. This is efficient, and it's been used for decades. However, it's not optimum in terms of information transmission and issues with updates of the available seeds in the seed room, for example. So that's calls for a better management here. Here, just a draft of the tool, a draft interface where you would see the different options to get the crosses exported to the database, the existence of a list of crosses you wish to perform before going to the field that you can upload, the printing ability. This is in the middle, the current draft interface where you would scan for each female and male the corresponding barcodes where you source the pollen and the flowers from, the name of the cross you would perform, and the saving option, the printing option, and the scanning option. On the right hand, I just wanted to show you a bit of what does it mean. So this is a BRAPI call for cross. It structures the information that are needed to be transferred between an app and the database itself. So technically, you plan a cross, submit the list to the tool loads it, you perform what you can obtain in the field in terms of flower and link that to the plan, and then get it back to the database right away and later for the seeds. With this, I will end the data acquisition section and we'll go briefly over several analytics the database offers, including BRAPI. So first of all, several analytics available in the database. So for any trial we have, you have a statistical descriptors section, which would provide real-time for any data loaded the descriptive statistics for a given set of traits using the ontology. It would show you the bar plots and their distribution, and also which are the accession content in any section of the bar plots, for example. Further tools are typically a correlation tool displayed here, where you can see multiple traits for a trial being put in relation. I don't think we have that on the slide, but there is also a principal component analysis and a k-mean cluster tool as well, which both work for SNP data, but also for phenotypic data now. More recently, we've been working on a mixed model analysis tool. So this tool aims at providing the user a combination of solutions for gathering data which comes from either from the field with other data types such as the year, the locations, and possibly the genetic data that's included in SolGS, and also in the future, why not environmental data. So the interface is still in the work, but what you can see here is you use the selection of what we call a data set, which is basically a user-defined set of information you want to analyze. And the selection of a given set of traits, which get composed here in the formula below, following classical mixed modeling. And on the right-hand side, you would see descriptive statistics of the data, but also which factor you can possibly add to the model or remove according to what you are investigating. So offering the user possibility to tailor exactly the type of analysis you want to do according to the data set you're dealing with.
Can I just ask what mixed model engine you're using?
So it's R-based, it's LME4.
Okay, thank you.
The Douglas-based package.
Mm-hmm.
Additional question on that?
No, I mean, I'd just like to, yeah, sorry, carry on.
Oh, no problem. Okay, so we can, so far it's a kind of a static demo, but we are happy to demo some of these tools. The mixed model is not currently released, so I would not be able to show it on the production database. So,
so if I could just point out, for example, the CIMMIT biometrics team already has a suite of mixed model functions for in R. You know, there's lots of scripts around.
Right. How is it called?
Just get in touch with Pancho Crosa or Juan Burgueño.
Yeah, because I think Jose Crosa has developed for a long time some nice tools, but some of them, for example, are Java standalone tools. So the challenge is not really the tool itself, but it's more the technology that is more or less easy to bring into a given system. So I hope I think they have an initiative to work with BreeDBase. Is that right?
I don't manage BreeDBase, so I don't know. I mean, it would be. I would be very reassured if you guys could answer that question.
So we're very interested in working with the SIMIT group. What we have done so far, we have actually worked with a SIP group with Hugo Campes and Bert de Bugs at SIP, and we're really working closely together with them to develop that tool. But the more the merrier. We should absolutely.
Well, I mean, that was sort of the idea of trying to set up a clearinghouse through the EIB module 5 or whatever it is, to actually, so that people would actually know what they're doing. Everybody knows what everybody else is doing. I see that that's not the case.
Well, yeah, I mean, I think in general we had some calls with EIB, but we have not come across that modeling at CIMIT. So
that's very disturbing. But anyways, yeah, carry on.
Excuse me. Hello, Lucas. Hello, everybody. So about the mixed model in general, I think is we need to think work in team groups. So not only, of course, that's it. We have a lot of experience. However, we need to think, work together with other organizations like Bruceños and Dr. Crosa, because we need to work together in order to have a very strong package, statistical package in this platform. So I think everybody's open to work together and improve the statistical analysis for breeders. So only is a one comment and suggestion that it's not with a one group. So we think we need to be open to in order to work with a statistical analysis.
Yeah, exactly right. So EiB from Eiger side has been leading this bioinformatics biostatistical analysis group under EIB Module 5. And their initiative definitely across all the different CG, different packages. This year, the focus will be diversity analysis, really try to see what packages are already available to benchmarking. And EiB has been hosting this Galaxy work flows, already integrated some of the packages from CIMIT bioinformatics group from Panqiu and Huang. They are part of that group. So I think Elisa, some of you guys from FIT and from ITA also came to Cornell, got exposed to the Galaxy as a framework with a lot of R packages contributed by Isaac, Lucas group also. I think we need to really talk to Kelly and EiB trying to coordinate maybe all the bioinformaticians get together, how this benchmarking before everyone go to different places to really replicate a reinvented view.
Yeah, and another thing that needs to happen is we need to understand what the workflows are in a selection pipeline, right? So we really need a lot of the tools that I see under development, are focused on research questions rather than breeding. So I think we need to really understand what analyses are typically required in the basic breeding steps and build those first. So we don't. I don't see that workflow clearly mapped out here or in some other packages.
I agree with Gary about this point. It's very interesting, your comment, and it's true, because not all the statistical, we depend on the stage of the breeding program. We need different kind of the statistical models or designs. excuse me, and different kind of the mathematical model that we support in order to resolve this question. So, but it's interesting to consider in your point, Gary.
Yeah.
Okay, yeah, thank you for those comments. I think they're super important, and
we should have put in a different topic and trying to cover that.
I think this presentation was not about pipelines, but about our database and the tools that are in there, and then how you stick these tools together into a pipeline is maybe a little bit of a different subject, I thought, but so we're not talking about it that much this time.
Right.
But I think, as you say, there needs to be a lot of discussion within the community. And we are not trying to go alone here, but we're working with people, and it's a big field, and apparently we didn't know about that one effort, and apologize for that, but we're definitely already working with other people, so it will be easy to include other people in our collaboration.
Yeah, so bring this to module 5.
Yes, bring these together. I think that would be super important. And if that is an outcome of this call, that's already a super good outcome, I think.
Thanks.
Okay,
thanks.
So maybe it's a good timing for this. Just after this slide, I wanted to talk a little bit about the breeding API and how it may contribute to solve some of the issues that just been mentioned. So briefly, the breeding API stands for the Breeding Application Programming Interface, which is technically data models which allow an easy transfer of breeding information between different systems. To do this, you can use different programming environments. So as of now, there is a breeding API version in R, which can come handy for statistical analysis. To tailor personal analysis when the one online are not fitting the user needs, for example. Other packages are implemented in JavaScript and also in Drupal. So the idea behind this is, of course, exchanging data and developing applications that can be shared across different breeding systems, such as BreeDBase, BMS, or B4R, for example. So really getting the community together in terms of use and development. So yeah, I just put here a quick illustration of how BRAPI can help systems to coexist and share and leverage information and resources. Resources are all available, of course, on the website, the BRAPI website, the R package at the specific URL. And the community is really growing. I was attending the last BRAPI workshop in the Netherlands 2 weeks ago, and we have more and more scientific groups and institutions which are showing interest for specific developments. And the good news with BRAPI is really that you don't need to implement the whole thing to get it to work, but solely implement what is actually needed from your instance, which makes it attractive to many. In terms of application based on BRAPI, our group has developed a set of them corresponding to different activities in a breeding program so far. So the main one, the initial one actually was the pedigree viewer, which is now implemented in BreeDBase, but I believe also in BMS from the last BRAPI hackathon. We have a new field map viewer, which is based on Google Maps and which includes GPS coordinates. So we hope that will support better new type of data, such as data from drones, for example. More on the genetic side, there is a map comparator, which handles high-density genetic maps. In Cell Genomics, it's implemented. And then more focused on the discovery or data exploration side. We have a trial comparison tool that allows you to plot multiple trials together for a given variable and see how data distributes across them. We also have a recent box plotter tool, which, as its name, just box plot any type of data from a BRAPI supported database. So in that case, it can be multiple trials, multiple traits, different types of design, and so forth. The BRAPI R package, as I mentioned, has got a lot of attention. And we have a graphical filtering tool which allows subsequent filtering of a given data set using multiple traits. For example, if you want to select accessions that perform with a certain amount of fresh weight, but also a certain amount of dry matter, that allows it. Another side, but important element to mention beyond the analysis is monitoring progress we made and see how users are acquiring the system we developed for them. So that's a part we pay attention to and we try to keep focused on, especially on the latest application development. We want to make sure we are reaching the audience. With that, I will thank many of the users, readers who are contributing to build our systems, and I will leave it open for some demo on elements you wish to be addressed or things you would like to see in a more specific way. Just let us know. I'll be happy to try to show you what the database is able to answer at the moment. Thank you.
Thank you very much, Guillaume. So we have about 5 minutes. Anyone have questions before we do a little demo? Comments, questions for both Lukas and Guillaume?
Hi, could you just comment quickly on the licensing arrangements? Is this free, freely downloadable and usable, or is it, how are you licensing it?
The props, I guess?
No, sorry, BreeDBase, the whole thing. Yeah, and the props.
Yeah, the entire code is available with a so-called MIT license. MIT is a very liberal license. It's the most liberal license that exists. Anybody can just download and use it in any way they want. There's no guarantee. Oh, I mean, there's no
user.
Anything goes. Yeah.
Okay.
And all the code is like that. And we're very careful in selecting backends and tools that have these licenses as well, so that this is possible. So we run on Postgres, we run on R, and so forth. So we don't have any proprietary tools in the entire stack.
Great.
I have a question. Not question actually, it's a request. So we are actually working with this EiB called this Enterprise Sample Tracking for this HTBG platform. So wherein, which is very similar to what you have for this genotypic job submission to Intertech dot kind of thing. So probably I request to start to have this group in this EiB working group, so that whatever the available tools can be leveraged with the EiB for the entire HTBG community. Thanks.
Yeah, thank you.
With Rajiguru's request, maybe worthwhile to do a quick demo on the sample submission and also genotyping service request.
Okay. So I will proceed for a brief presentation on how the database handles genotyping data. I will just get you started with the home page, where you need to log, so that you can get the permission in terms of data edition or upload that are linked to your account. So the home page presents you the main menus to deal with all the components. Under Manage, there is one section that addresses specifically tissue samples and genotyping plates. We differentiate these two so that you have genotyping plates for standard tissue sampling acquisition for genotyping into PCR plates, basically, and tissue sample is more like tailored when you do a specific targeted experiment, such as RNA-seq, where you may sample several leaf flowers at different time points, not in a particular plate frame, for example. So I will just pursue with the genotyping plate section. What you can see here is an interface which is maybe similar for those of you who are familiar with the system. It's similar to the interface we have to show field trials. So basically, under different breeding programs folder, you would find the related data stored there. You will have to pardon our mess because I'm demoing on the test version of the database, so that I can edit as I want information without disrupting anything. Here we see, for example, the Cassava database, where we have different programs. Under each of those programs, you can structure information as you wish. In that case, you see folders that might be created to store a specific set of information under here several years, for example. But you can also have standalone experiments when it's just about one and you don't really need to have it under a given project. Here, I will show you what is already stored as data. So I double click here. And you obtain a page which is specific to the plate being loaded. Well, the page gives you several information on the type of data. So it's a 96 plate by opposition to a 384. You're doing DNA. It provides the platform you selected. If it's submitted online, in that case, it's not submitted online. And you have the ability to download the formats requested by different providers. So far, Intertech and Darkseek. So if I double click here, okay, why is it doing this? So here, just to exemplify a plate, I loaded. You have the plate layout below. For each of the samples, you have several information displayed, the ID of the sample itself, the accession it comes from, and the source plot from the original trial it comes from. So all these are hyperlinks and can be clicked to get, and you will get sent to the corresponding accession or the corresponding trial the data comes from. Before going further, I just wanted to show you, clicking on the download of Intertech, you would get the flat file that corresponds to the exact template required by the provider. So namely the sample ID, the plate ID, the well location, the corresponding ID of the barcode you've scanned when you were in the field, the name of the plate, and comments if needed. Is there questions?
So this can generate from any, let's say, 15 plates at a time.
You can. So currently, maybe I should go one step back. So this is a final result. I didn't know how much time we have, so I just wanted to show you what can be obtained. But if I go just one step back here, when you have two processes, so the first one you want to look at is uploading the plate. So that follows the procedure I described earlier, which involves the coordinate app from PinoApps. So when I reach this step, I assume to have with me the files I obtained from the data collection in the field using coordinates. And from there, I will start a workflow just to upload. So namely, it will tell you what you can upload from this interface. So 96 to 84 plates, more description on the provider and so forth. If you click next step, it will require you to specify which interface, which provider you wish to submit the data to, which project you are affiliating these plates to, the current ID of the plate, the name. You can specify the type of tissue you're dealing with, the affiliated organization, the location of the plate itself, the purpose of your work, and then it will take you to the next step where you have to specify if you want to upload data using coordinate, or we made also possible that people use Excel in case that they do not have a barcoded field, they can still have a way of dealing with this. If I select custom coordinate Android app, for example, it will show you which template is expected. So this is the template from the app. Let me see if I can
Yeah, just hold on a second.
Yeah.
Yeah, you can continue. So for people on the line, the webinar for today is officially adjourned. Thank you so much for joining. And for anyone else you're interested to see more and you want to have more discussion, and you can stay on the line. Okay, go ahead.
Okay. So I'm about just, I think I can try this plate here. I will try to load here. I will take the risk of doing a live demo and load that thing. So I selected my coordinate template, and now I reached the final step of the pipeline, which is technically we would be able to submit directly to a genotyping platform through BreeDBase. At the moment, we do not have, I think, any client with whom we can immediately interface, although we know that people are working hard on that. But so what I want to say here is no online submission yet, but at least it can be stored to the database.
Guillaume, I have a question here. Can you submit a group of plates here?
For now, you can only submit 96 format plates.
Right.
If you have
one at a time, or you can do many plates based on how long they are long.
Following coordinates, we have single files, and we want to have this batch upload. It's not available for coordinates. If you do non-coordinate data, such as Excel, you can load more rows, more plate data at once. Yes. This is something we want to prioritize now. We have a large adoption of this tool, so people are requesting this.
Okay.
And also for the submission to the genotyping facility, you can organize your individual plates into folders. And I think currently it's not implemented, but the idea is then that you can also submit a folder, and it will process and send all the files.
Yeah, I think a folder would be nice because that's what we normally do. We have like a group of group of plates, and we want to submit, let's say, 100 plates at once.
Right, exactly. There are very large numbers of plates sometimes because, So it's important to do that in an easy way.
Okay, so it proceeded. I loaded the plates. I close it, and now I can refresh my page and search for the. Oops, sorry, this is refreshing. I was too fast, sorry. Now I can search for the plates I just submitted, which is right here. So you will see, again, the same interface as I briefly described earlier. The associated data to my plates, options to download, ability to generate a barcode if you want. So most of the platforms require barcoded plates so that they can process them. So there is this option here. You can decide to generate a barcode specific to that plate before shipping. And here you have a sample set of what the plate can contain. The blanks are also highlighted. In that case, you see in this orange B2, well, it's a blank. Again, if you want to reach the trial where the data comes from, it's easy. You click the source plot, and it will match up the given tissue to the trial it comes from. It takes me to this page, and it will match up the trial name where data is being collected. Okay.
So I understand Intertech doesn't have online submission. So suppose for start, how do you continue on to submit your?
Yeah, I think with that it becomes now possible because Gregor and Putek have recently implemented the BreeDBase vendor calls in their interface. So we are having conversation with Dart to see how we could possibly
integrate. Currently, you need to go
get
to
a better integration. That might not be right now, but that's hopefully in the near future. Yeah.
Okay.
Okay, and the last phase would be once you get the data back, you have the second section which is called Upload Genotyping Data, which is an online interface where you can submit the data you obtain from your tissues and which will hold the same identifiers. So it will plug the corresponding genotypic data to the samples to the trial, essentially.
Guillaume, can you add the shipping information if you ship a plate?
I guess that would be linked to the user profile to get credentials to submit data or not. We could create a particular status for the user to be a plate submitter, for example, so that it's restricted to people who do have accounts to the genotyping platform and avoid errors on that side. So yeah, I think it's feasible.
Yeah, currently any submitter can submit plates, but maybe that needs to be more restricted. Yeah.
But yeah, there's no the web interfacing is a dead end at the moment.
Right.
So there's no risk.
Yeah. right, exactly. But
don't worry.
Yeah, we have to work with G a little bit on that interface, and of course, we have to work with you again a lot more to implement it on this side.
Yeah, last Friday I sent everyone integrated genotyping service workflow. There's a diagram T and I work together. I think we should follow up on that and have some prioritization to see what we want to do. More streamlined submission and shipping checking and data retrieval. Yeah.
So Lucas, I have a general question. So the thing is, like, every breeding program might have slight different workflow. So by looking the interface and the web database you have, it seems like it can do everything, but sometimes each breeding program might need like slightly customized interface. For example, like the like a manage menu, and each breeding program might require like different managing menus. So I just don't know how easy to customize it. Or
well, if it's just an interface question, it's extremely simple to customize because the way we've implemented customization, so we can each website can actually have its own skin, and so you can adapt the interface really easily. If it's more of a back-end thing, though, it's a little harder to have customization.
Yeah, I think probably not back-end, mostly it's front-end. So the way we work is, say, for example, the 5 crops supported breeding insight can just get breeding insight development team to help with customize, right?
Yeah, for example, yeah, of course. Well, I'm speaking for them, but Tim is okay with it. So.
Yeah.
All right. Given the time, I know people calling in from different parts of the world. Very late for some of you guys.